Okay, so the cell cycle refers to any kind of reproduction of a cell. So all the cell cycle starts with is all starts in the same thing. So interphase is the first stage of the cell cycle, regardless of what happens in the cell. And two really important things happen three in order. So the first thing, in order for any cell to grow, we have to have lots of proteins. So protein synthesis is the first stage of any cell cycle. We have to make lots and lots of proteins. Again, just because we're here already, let's just add a bit more detail to this. Protein synthesis, what we're actually talking about is transcription, which is copying the code, and then making sense of the code is translation. I know Dominic wanted me to do transcription and translation in a bit more detail, which I will do. We'll do that more because that's a paper 2 stuff, so we won't focus on that now. Once we've finished protein synthesis, I need to replicate the DNA. So DNA replication is the second stage. And what that actually looks like, which is what lots of people get confused with, is my chromosome originally will look like that. It will just be a single strand of chromosome, okay? When it replicates, that's when it looks like that. Okay, which is just 2 strands. Those 2 strands are actually exactly the same, but it's just a copy of each other. So I've just copied the same strand twice, because then what happens in cell division is what I'm really actually doing is splitting these 2 in half. One goes to one cell and one goes to the other, but I have to replicate it first so each cell can have an identical copy of their genetic material. Once we finish DNA replication, the cell then gets bigger in size, in particular, the subcellular structures. You could use the word organelles instead, but subcellular structures replicate. Okay, you could give an example of this. It could just be e.g. I would probably say the best 2 examples to give would be ribosomes and mitochondria, because they're the most common ones for that. So that happens in all cell cycles, okay? So regardless of what the cell division is, the first stage of all cell cycle is interphase, and these 3 things happening. So this is before mitosis, before meiosis. Once we finish then interphase, that's when we go on the second stage of the cell cycle. Don't write that. Is nuclear division. So that's where we're dividing our nucleus. And there are 2 examples of nuclear division. One, we're going to do maybe today, but certainly we're going to start in your year 11s if you've not already done it, is meiosis. That's a paper 2 content. The paper 1 content is mitosis. So the 2 different types of nuclear division, or 2 different types of cell division, which sometimes they are referred to, are meiosis and mitosis. Just to simplify it really quickly before I do either of them in more detail. Meiosis is the production of gametes. A gamete, as hopefully you know, is a sex cell. So that is how we produce sperm cells, egg cells, and pollen cells. So there are 3 types of gametes that are produced: egg cells, sperm cells, and pollen. Mitosis is the production of body cells. Okay, any questions? Right, let's now look in more detail. I don't want that. Let's look in more detail now then, please, at mitosis. So I'm just going to talk about mitosis now in a bit more detail. So I have my cell, and I'm just going to simplify it. So here I have So here is my cell at the beginning of mitosis, okay? This cell has 4 chromosomes. A common mistake that students often make is whenever we're talking about the cell cycle, you always say 23 pairs. That only exists if it's a human cell. Remember, this is biology, not human biology. So don't say 23 pairs unless it's talking about a human division, which it might not be. So this cell here, just a random example I've chosen, has got 4 chromosomes or why is it being annoying? 2 pairs. The reason it has 2 pairs is because you have one set which is from your mother, which we call a maternal chromosome, and then you have one that comes from your father, which is your paternal. So that's why they come in pairs. 4 chromosomes, they come in pairs, your maternal and your paternal chromosomes. What happens then is kind of, you'll notice in my next diagram, there are 2 big differences. Okay, the first thing you can see, and it could ask you to describe the difference, you'll see the nucleus has disappeared. So actually the first stage of mitosis is that nuclear membrane completely breaks down. That allows the chromosomes to be freed for the whole cell, which is really important. And then the key thing, as you can see here, is my chromosomes have now lined up in the middle of the cell. And then the final stage is what's going to happen is each chromosome now gets split in 2. So effectively, as I kind of drew earlier, we draw a little line down the middle of this cell and split it into 2 different cells. So then what ends up happening is we then end up producing one cell, we end up producing another cell, and it looks like that. So I've now produced 2 cells. We no longer have that X-shaped looking chromosome because I've split it in half. We've produced 2 genetically identical, and I would always use the word genetically identical rather than just saying identical. Genetically identical cells. These cells, because they're genetically identical, the main use is for growth and repair, in particular growth. So that is how a cell will grow. Okay, we're now going to focus on a bit of an application. So how does it actually apply in real life? We're going to talk about from fertilization, so the very beginning of life, and then we're going to link that to stem cells. So the next one we're going to look at here. So what I'm going to have here is I'm going to do fertilization is kind of my title. And this one, I am going to talk about human fertilization, so I'm going to link this to a human. So this is only human related. Fertilization starts, of course, with the sperm in my egg cell. So I've got my sperm in my egg cell. The definition of fertilization is the fusion of the nuclei. So the sperm cell contains a nucleus, the egg cell contains a nucleus. What happens in fertilization is these 2 nuclei fuse together. And that is what fertilization is. The only thing that comes from the man is the nucleus. Every single organelle that exists in your body, so we're all men in this room, every single organelle that exists in our body has come from your mum. And when you have a child in future generations, every organelle your children has come from the mum. Because the only thing that gets passed on from the man to the woman is the nucleus. Or a man to the child is the nucleus. And obviously that has, that makes up 99.99% of what they're gonna be. but lots of people have something called a mitochondrial disease. You can only inherit a mitochondrial disease from your mum. If we have mitochondrial diseases in this room, we cannot give them to our children. It's irrelevant because it won't get passed on to the child. The only thing that goes from the father to the child is the nucleus. Okay, so this first cell here, apologies for that, is a zygote. So this is called a zygote. So that's a fertilised egg cell. And this zygote, of course, contains 23 pairs of chromosomes. This zygote will divide by mitosis. To form two identical cells, two genetically identical cells for growth and repair. This is the very first stage of, and this is called an embryo. This embryo then contains all of your genes. Which means what we can do, because it contains all of your genes, and at the moment, the embryo is undifferentiated. So because of those two properties of an embryo, an undifferentiated cell that contains all of your genes, that enables me to use an embryo as a stem cell. Okay, so the next kind of subtitle then is, what is stem cells and how can they be used? I'm going to give some context. I'm not just going to go through all the basic stuff. I'm going to add in more detail. In every single year, IVF, which is a process you should have learned about now, in vitro fertilisation. In vitro fertilisation, the aim of in vitro fertilisation is to produce lots and lots of embryos. Okay? So in vitro fertilisation, sperm fertilises an egg cell. We produce lots and lots of embryos to then potentially be used to produce a child. These two might go into the mother. We say they might go to a surrogate mother. So these two are implanted in the womb. These three are not. Because otherwise the woman could end up having 5 children, which she might want, but I'd say it could be quite painful. So we've now got 3 embryos, which if nothing happened to them, they would just go to waste. So instead, what we could potentially do is use these as embryo stem cells. And because these embryo stem cells are undifferentiated, they could become any cell or organ. And the main examples they'd expect you to know about in terms of any cells or organs they could be used for. So they could be used to make an organ for organ transplant. For your specification, there's 2 particular organs they would be talking about. You could use embryonic stem cells to make a pancreas to treat people with diabetes type 1. Or you could use embryonic stem cells to make a kidney to treat people with kidney failure. So there are 2 main examples they could talk to you about in terms of organ transplants. The second use then could be to make nervous tissue. And the main treatment for that is paralysis. So they are the uses of embryonic stem cells. Lots of people are, of course, against using embryonic stem cells. Because there are significant ethical considerations for using embryonic stem cells. What do we mean by that? well, all of those embryos has got the potential for life. Be it that we are putting some of them back into the woman, clearly they've got a potential for life. The other issue then with these embryonic stem cells is they do have a risk of rejection. Because these are embryos of somebody else that are then being used to make a pancreas or a kidney for me. They are not my cells. So they're going to be rejected. Well, they could be rejected. They might not be, but they could be rejected. So two main considerations for using an embryonic stem cell are the ethical considerations and the rejection. Okay? However, what I could do then to remove the risk of rejection, so to prevent rejection, I could use something which you might have learned about recently, is I could do something instead called therapeutic cloning. And in a simplified way, what therapeutic cloning is it's turning my cells into any cell. Any human cell, I should say, but any cell. So that's converting a skin cell into a kidney cell. So I could take one of my skin cells and make a new kidney for me. So that's using therapeutic cloning. And what that looks like is this. So in order to do therapeutic cloning, I need 2 cells. Okay, so therapeutic cloning, I need 2 cells. I need an egg cell. Obviously, funny enough, being a man, I don't have any egg cells myself, so I need to have a donated egg cell. But I can get those relatively easily. They can just be removed from a woman's ovary. And I need one of my cells. So this cell is the cell I want to clone. Okay? And as I said, it could just be one of my skin cells. Because remember, all of your cells contain all of your genes. I'm then going to remove this nucleus. So I take the nucleus out of the cell I want to clone. I'm then going to take the nucleus out of the egg cell. Which means I'm left with an egg cell that looks like that. This is called an enucleated egg cell. Because the nucleus has been removed. What I then do is I add the nucleus of the cell I want to clone into the egg cell. If we go back to what I was talking about a second ago, why do we do that? Because the egg cell also contains all of the organelles you need in any cell. It contains huge amounts of food store, so that cell can then grow and divide. This now enucleated egg cell now contains the correct nucleus. I then stimulate this egg cell to divide. Usually give it a very small electric shock just to kind of kick it on its way. And this will then divide into an embryo. But it contains the nucleus of the cell that I originally took it from. So now, if I look at my diagram here, I'm in exactly the same position that I was up here. I've got undifferentiated embryonic stem cells. Exactly how I was before when we spoke about fertilization. The difference is now, though, these undifferentiated embryonic stem cells have now got my genes in them. They are my cells, which means they're not going to be rejected. So this then means I can use these. So I could, so if you've just done kind of kidney transplant, kidney failure, I could use these now to make a kidney, which allows to have those. Ethan?
How do you make sure the cells differentiate into kidney cells?
So, much like when you've done plant hormones at the moment, every single cell needs to receive a different hormone. So I would give it a particular hormone. There's another type of chemical, you don't need to know the name of this, but it's called a cytokine, which is a chemical messenger. If I give that cell the correct message, it tells them to turn on this gene to become this cell. If I want it to become a nerve cell, I'd give it a different chemical messenger, a different hormone or a different cytokine, and it becomes this cell. That's why at the moment when you're going through puberty, some of your cells are turning into hair cells, some of them are turning into muscle cells because they're receiving a different message from a hormone or a cytokine to turn into that. Any questions? Okay. That then is a bit of cloning, bit of stem cells, bit of mitosis. We're going to move on to looking at meiosis. Should be doing it in lessons, next couple lessons soon. I think we'll call that a day.